Welcome. Happy Friday. I'm Dr. Grip from psychology and also the behavioral neuroscience program. And we have a couple other faculty here from the program today. I talked to some chem professors several months ago, and I thought it'd be kind of a cool idea to try to build some more bridges between departments. We've done a joint seminar with biology earlier this year, and we're looking at doing that again in the fall. We're looking at having one with business school, and who knows what's next, computer science, maybe something like that. But maybe rotating this around, keep it going. So finding a guest speaker, typically somebody fairly local, to come in and talk about their research. So we can get to know them also because they're local, making connections. But for this particular joint between BNS and chemistry, having somebody that literally bridges those two areas, much more chemistry, but also I'll let you explain it all. It's really sci-fi sounding, really fascinating information that you're going to be covering. And so I'll read the background here. Dr. Amani Hariri is here. She completed her PhD in chemistry at McGill University, where she focused on using single molecule fluorescence methodologies to optimize the assembly, I stole this from the website, of artificial DNA nanostructures to enable their applications in drug delivery and medicine. In 2016, she started her postdoc at Stanford University in the School of Engineering in the lab of Professor Tom So. I didn't like that part, where she was an instructor in the School of Medicine. Very cool. In the So lab, she explored the design of novel materials, structure switching probes, and optical devices for early detection and personalized treatment of diseases, and the development of real-time aptamer-based biosensors that are capable of continuously measuring biomolecules in vivo. You can see the bridge over to behavioral neuroscience there. The Harari group at UBC, it just moved there very recently, combines expertise in molecular design and optics to engineer novel single molecule tools and transformative platforms for monitoring and diagnostics to help unravel some of the grand challenges of biomedical research. The group is pursuing the research areas of optical biosensors, novel single molecule based diagnostic platforms, and aptamer based switches for multifunctional stimulus responsive nanosystems. What? My research is so boring. So everybody, please welcome Dr. Hariri.
Thank you. Yeah, thank you, Jeff, for the introduction. And I'm very sorry about the Zoom thing. So sorry for the Zoom people who are seeing the next slide now. I guess their dopamine will not be the same because they're going to see the, yeah, so I guess you summarized where I come from and my background. And today, basically, I'm going to talk about, because I just started at UBC like a month and a half ago. So I'm setting up my lab. And what I'm going to be talking about today is my project that I worked on at Stanford. And this is more developing aptamer-based optical sensors for the real-time detection of neuromodulators in the brain and other also small molecules in complex matrices. Now, just to first address the question, why real-time biosensor? I'm going to start like the engineers in our lab used to start with, is an airplane. So an airplane being like a sophisticated machinery in order to make sure like the airplane is safe for the passenger that is taking. The engine of the plane has more than 30 sensors that will track in real time and monitor all the time the different parameters like the temperature and pressure and resolution. So you actually could detect a failure early on and prevent a problem from happening. So we do that for an airplane, but surprisingly, we don't do it for a more complex machine that is our body. So for the human body, what we usually do is go to the doctors once a year And that's if we are careful. To be more realistic, we actually go every 5 years maybe, or when things go wrong. So when it's probably too late, or when a disease became like a bit uncontrollable. And the reason, and unfortunately, we're not actually monitoring our system very frequently to be able to get to detect these events early on and be able to predict a disease before it happens. And the reason why is we don't have the convenient sensors, the convenient tools that would enable us to more frequently monitor at the molecular state our system. Now, more recently, I mean, in the recent years that it came by, we started having more devices, and these are to mainly monitor our physiological state. So half of us probably are wearing an Apple Watch or like a Fitbit. So I've been wearing that actually for many years now, since they released it, and I have like established a historic baseline when I was healthy. So you could see that my resting rate, say here, was 59 for many years before January 2021. After January 2021, actually, I was diagnosed with non-Hodgkin's lymphoma. So if you could see my resting rate, it actually jumped to 74. But that was few months before they diagnosed me with the condition. So my monitor was actually giving me an alert that, you know, your resting rate is trending higher and is above what usually historically was your baseline. So this is the kind of data that we could collect from a real-time sensor. But unfortunately, I mean, this could have been also the flu. This could be COVID. This could be anything. These monitors, like monitoring only physiological state, has low diagnostic specificity. So what we ultimately need to be able to detect a disease, a life-threatening disease, from a mild disease, or distinguish between these two, is more a sensor that would monitor our biomarkers, like more biomarkers in our body. But this is difficult because this sensor has to operate in difficult conditions, like in blood, in like tissue, and it has to be specific and sensitive. And so far, what we were able to get is like the glucose meter, and that everybody is familiar with. It has really transformed actually the life of diabetic patients, because it enables them now to manage their diabetes. But unfortunately, the chemistry of it is very specific to glucose. So it's an enzymatic reaction that is specific to glucose, and it's not generalizable to other molecules. So what we need are sensors, are like platforms that are more generalizable to other biomarkers. And this is what Tom and Kevin 10 years ago in Santa Barbara have come up with. This is like they thought almost now 10 years ago about like an electrochemical based sensor to devise a generalizable approach to measure drugs in blood of actually live animals. And at the heart of the sensor is a DNA aptamer. And DNA aptamer, if you're not familiar with it, it's a single-stranded DNA, and it binds to the target specifically, just like an antibody binds. And it actually, like in addition to what the antibody does in terms of the binding, it flips its conformation when it binds to the target. So what you could do is add a label. In this case, they added a redox label that is indicative of current. So when the target binds, it changes the conformation, and the current would change. So they could actually correlate the current change with concentrations. I'm going to show the video that they usually show is the first experiment that they did, and how they developed the platform is by injecting doxorubicin. It's a chemotherapeutic drug. So they inject it in a live rat. And what they do, they draw like small amount of blood and put it in a microchip. And this is like the medic device. So it's coated with gold, like I showed in the previous slide. And because blood is complex, it has lots of like cells and proteins that could affect the surface and could affect the binding ability of the aptamer. They actually created a sheath layer of buffer in between the surface and the blood, but the drug would still be able to diffuse onto the surface and bind to the aptamers that are grafted on the gold surface. So now if you flow in a low concentration of doxorubicin, few aptamers will bind, and then you see a small increase in the current. But when you increase it, you see more of these aptamers will switch their structure, and you would see a further increase in the current. So if we zoom in on the aptamer, this is before the target binds, the current is low, and after the target binds, the current increases, and what they do is measure the differential between these two, and then actually generate a plot like this one, a real-time plot. So we could actually monitor the increasing and decreasing concentration of doxorubicin in like the low to mid micromolar range. So we could actually sensitively detect doxorubicin within the therapeutic range of the drug. And not only that, it's actually specific to dogs. So even if you put it in the presence of a large excess of other molecules that have similar structures, so this is a huge excess, you only would see the signal when dox is there. So it's very specific, and it has real-time response. So the on and off kinetics are super fast. So it could detect things really quickly. And we don't only detect molecules in vivo, they were able to actually add a feedback loop system. So in this case, they're also defining a profile for the concentration of the drug in vivo in a live rabbit. So what they do is that set a certain concentration profile. So I would tell the sensor to increase the drug concentration to a certain level, keep it there for about 20 minutes, and then go back to baseline or to zero. And this actually is useful for drugs that have narrow therapeutic range, so that it actually stays at a certain level throughout the treatment and be more effective. So basically, that's what they've developed so far. They have done that for doxorubicin and kanamycin, so they could detect drugs in blood. But our body is more complex than that. And the most complex, and correct me if I'm wrong, I'm not a neuroscientist, but I guess the most complex organ in the human body is the brain. And in order to understand how the brain mediates its functions, we need more tools to measure the fast release of small molecules like neuromodulators with high sensitivity and high specificity. And neuroscientists around the world, and here specifically at Stanford, have established that specifically dopamine and serotonin, the neuromodulators, have a huge effect on behavior, and they play most likely a major role in depression, addiction, obesity, and autism, probably. So the current state-of-the-art techniques are limited to microdialysis, FSCV, PET, and fMRI. Now, they have many advantages, but they do have also their drawback that is limiting the field and limiting us from understanding the circuitry of these two neuromodulators. So some of them have slow temporal resolution, some cause tissue damage because they're pretty big. Some are not generalizable. Some have high background, and some have poor specificity, so they cannot distinguish between, say, dopamine and norepinephrine. And what I discussed previously is the MEDIC device, a device that could be generalized to other molecules than drugs potentially. But the issue with this method is that it detects drugs that are in the micromolar to millimolar range, so they're high concentration. And the biomarkers or neuromodulators that we're trying to detect are at low concentration, so in the picomolar to nanomolar range. So we need a more sensitive approach, and we need an approach that could last for a long time without fouling, without affecting the surface, and that could be easily multiplexed and implanted in the brain of a mouse. And what we thought of is, instead of using EChem, instead of using electrons, we could use photons. And I'm not sure if you're familiar with a fiber optic, but people have used it in optogenetics, for example, to stimulate neurons. But in this case, we wanted to use it to measure molecules. So we wanted to collect photons and correlate them to concentration of biomarkers. So in this case, we wanted to create a bundle of different fibers. Each of these fibers will have a different switch, and each of these switches would have a different fluorophore at a different wavelength. So this way you could create like a miniaturized sensor that could be implanted, because the size of the fiber is really tiny, and you can implant it in a specific region of the brain and monitor different neuromodulators simultaneously over time. So we actually built this platform, and it has many components. The first component is the hardware, and that's the detection box, basically, or a detection like a module, to which through which we will collect the photons from the probes that we're going to be using. This detection system will be connected to the device that will be implanted, and that's the fiber. So we will optimize the fiber to maximize the signal that we will get from our probes. And finally, we want to develop ways to attach these probes on the surface and develop ways to make these switches. How do we select them? How do we make them? So I'm going to go over the different parts like that form this platform. So the first one is the hardware, and that, in collaboration with the Vikovic lab in the electrical engineering at Stanford, we basically created this box. So from the size of a room, we actually we had like huge lasers, huge like fancy detectors to detect the photons. But now what we did, we miniaturized it to a small box that could be actually transported to other labs, like the neuroscience lab, to measure molecules. And this actually box will embed the detector, which is a single photon counting module. It will embed the laser and all the optical components to collect the photons from the fiber. So this box will be connected to the fiber. The fiber is like an evanescent sensor. So if you're familiar a little with optics, what happens is we shine the light at a certain angle above the critical angle, and it will undergo total internal reflection within the fiber optic. So the light is confined within the fiber, and then we taper it. So we thin it down towards the end to make it first smaller and to actually maximize the field at the tip of the fiber to be able to excite the molecules at the end of this fiber optic. So we did that, and we created profiles that are reproducible to create each time the same device and generate the same signal. And then we needed to attach aptamers, to attach the probes on the surface of the fiber. So what we need is techniques to attach them specifically on the surface and to passivate the surface to make sure that we minimize anything non-specific to bind to the surface. So we want to minimize the non-specific binding. And for that, we got the best result with a PEG monolayer on the surface. So what we do, we clean the glass fiber, and then we aminosilienize it, and we add a PEG with a succinimide at the end and with a biotin molecule. And this biotin will bind strongly to a protein called streptavidin. Streptavidin has 4 binding sites. One site will bind to the biotin on the surface, and the other site will be available for other biomolecules to bind. And this is how we will immobilize our aptamers. We will modify them with a biotin, and they could actually bind specifically to our surface. So in the absence of, for example, streptavidin, if I don't have this protein here, you see that I added the DNA and I don't see a signal. So that's the control that we did. And then if you add all of these components, you'll get the signal on the surface. So our signal is specific and our non-specific binding is minimal in the absence of one of the components. Now, additionally, if you add the fiber, just to note, like if you add the fiber in the presence of like lots of fluorophores in solution, you would barely get any signal going through the fiber because it would detect only things that would be on the surface. It acts like a near field sensor, and that's to like remove everything out of focus or in solution. So it would focus on whatever would sit on the surface. So the signal between these two will be almost similar. So now what we have done is really optimize this backend. We optimize the optoelectronic system, we optimize the fiber and the surface. We still have the aptamers. And our group have developed a technique called the particle display methodology, and that's to actually create these specific aptamers that would bind to a target specifically. And to do that, what we start with is a pool of DNA sequences, a library of DNA sequences. And we isolate each DNA sequence with a magnetic bead by creating a water and oil emulsion. And then we amplify these beads to create multiple copies of the same DNA on this bead. And at the end of the day, what we are creating is a library of these multiple copies of DNA. What you could do is challenge it with a target. You label your glucose, your dopamine, your serotonin with a fluorophore, and you challenge this library with the labeled target. So at the end, what you get, the best binders will actually have a higher fluorescence because they will bind more tightly to the target. And you could use FACS to sort through these particles and pick the ones that have the highest fluorescent signal. So afterwards, you could amplify the sequence and you could sequence it and get the best binders that you have. You could repeat the process to get a higher affinity aptamer or a more specific aptamer. Now, this process is, I'm not gonna lie, is tedious. Like it takes really like a long time to do. So what Diana just did is actually hack a DNA sequencer, and she's able now to select the binders directly on the flow cell of the DNA sequencer. And that's like a more recent approach that the lab have published. So we have different techniques to actually select these aptamers. The only issue is that we're selecting for them to, we're selecting those aptamers for them to bind the target specifically, but we're not selecting them to flip and switch their structure. So how do we actually get them to switch? Just to like the ones that Tom and Kevin have done before, doxorubicin and kanamycin, were inherently structure switching. So yeah, I'll be honest, I guess they were lucky. They got them to switch, but they don't know why. We don't know why. But what we want to do is make every aptamer that we select through these techniques a structure switching aptamer. So what we did with Brandon and Ian, we're taking an existing aptamer, label it with a fluorophore, and then we run a competition reaction between the target and a short complementary strand that has a quencher. So whatever is displaced by the sequence that is displaced by the target, so that the target outcompetes for the binding, we link it through a poly-T or a polymer, and then we create an off signal beacon. So a beacon that has an off signal because the quencher and the fluorophore are in close proximity. So when the target binds, it displaces this blue strand here, and the fluorophore will be away from the quencher and will give you an on signal, a fluorescent signal. So we did that with the ATP aptamer. The ATP aptamer is the most established aptamer. So you could do whatever with the ATP and it would work. So that's your positive control. We actually did this strategy, and then upon adding ATP, you see that the higher the concentration, the higher the signal. So we are creating this switch. And then not only that, we could also like tune the switch. So if you want a better affinity aptamer. If you want a faster aptamer, you could actually tune the length of the loop, you could tune the displacement strand, and then tune by that the thermodynamics and kinetic property. So if I need like a more sensitive aptamer with a different binding affinity, I could now actually tune the KD. I could also get a faster aptamer. If I have a slow aptamer, when I generate it, I could actually tune these two knobs and get a faster aptamer. So this we all did, like we did it with the ATP, but I was able to also do it for dopamine. So for dopamine, we're able now to get a switch that has okay affinity. And then initially the aptamer was actually very slow, which was a mismatch with the fast release of dopamine in the brain. So we used this strategy to make it orders of magnitude faster. So now we have a very fast aptamer. So now we have the different components coming together. We have the optoelectronic device, we have the fiber, we have the switch, and we could actually start running the experiments with the dopamine switch, for instance. So we put the fiber in a microfluidic chamber, and in buffer, in artificial cerebrospinal fluid, we inject different concentration of dopamine, and we're able to see increasing and decreasing concentration of dopamine from nanomolar to micromolar range. So it could span like a high range of concentrations. We still need to optimize in the low range to detect like endogenous release, but it's a good start. It also is a very like reversible sensor. So it actually could be regenerated for many, many times. Now, I could only do 24 hours. Maybe you're younger, you could actually do more, but that's actually the minimum that we could actually do. So we would inject back and forth dopamine and buffer, and we'll get it to recycle over and over. So the aptamer is robust on the fiber. We could repeat it for at least 24 hours in buffer in ACSF. And is it generalizable? We did it also for serotonin, and now we were able to get a switch that can go down to single-digit nanomolar with really high, with really fast kinetics. So very fast sub-second K on and second in the K off, that is in the seconds range. So now what we got roughly is like sensors that have good temporal resolution, good sensitivity, that is comparable. I mean, we would say it's still not to the point of genetically encoded sensors that are nowadays actually the state of the art for optical detection of dopamine and serotonin more recently. But what we are trying now is to start like the collaboration with Rob Malenka at Stanford and his lab, where we want to implant this fiber in live brain slices. So what they do is they cut the brain and they keep the slices alive in a rig in like artificial cerebrospinal fluid. So we insert it we want to insert it in the tissue and in the slice. And either in the striatum or like in the nucleus accumbens, we want to figure still like in which region we want to insert it and bath apply MDMA or other drugs to evoke the release of neuromodulators like dopamine and serotonin. So to benchmark also our technique, because it's still new and we don't know like whether it will give the right results, we want to have a reference point. And that's, we're using FSCV to actually get the optimal condition on where we want to insert our probe. So we did like some electrical stimulation to get like where actually we would get the best dopamine release. And we're actually bath applying amphetamine to try to see like what concentration of the drug should we actually apply to get the highest concentration of dopamine released in ex vivo setting. So eventually when we will achieve this is still preliminary. when we achieve the ex vivo measurements, we want to insert it in the brain of a mouse and be able to eventually, this is our dream, to place the fiber in the brain of a mouse, and following a behavior, we want to measure the release of these neuromodulators. So this is like really summarizing the end goal of our experiments. If we zoom in now, you'll see like the fiber inserted in a specific brain region, and again, like through the aptamers and the fibers and the photons, we want to measure the release of serotonin and dopamine, and eventually extend it to other molecules, small molecules or neuropeptides in the brain. So yeah, this is to summarize everything. And we also, like, this is like the last two slides, I promise. Like we also are trying to detect other molecules. So we were able to get switches for cortisol. So we are doing that for cortisol in buffer and in human plasma. So what I want to do now in my lab is really expand it beyond neuroscience and try to detect things, to like miniaturize these sensors, put them in blood, and create these like smart robots that could go and measure things in the body. And on that note, I'll just like to thank my supervisor at Stanford, Tom So. Yeah, he's been like really crucial for everything that we've been doing so far. My collaborator, Dovikovic, Malenka Lab, and everybody who was involved in this project. Yeah, and thank you for being here on a Friday afternoon. I know it's hard, but yeah, hopefully that was clear. Yeah, thank you. Yeah.
I noticed a lot of the drugs that we were testing are synthetic drugs. Were you doing it on any natural drugs?
Sorry, I did not.
Like MDMA and
Yeah, we did not try any natural. No, I think it's just like we tried cocaine also, but we only tried these drugs. Yeah.
Okay.
Oh, okay.
I had a question from a participant on Zoom. They said, you mentioned that the end goal of the technology is to deploy in animal models, but do you foresee this being implanted in humans, and for what specific purpose? But I think they might have posted that right before you covered that on the slide, but if you wanted to.
Yeah, I mean, eventually the goal is, because here I did mention the genetically encoded sensors. And these are, like I said, the state of the art for mouse now, and like other kind of fish and other animals. But it's really hard to do that, like the virus expression that is involved in this process. It's hard to do it for primates and for humans. So that's why like this technique is still limited to animals. And if our technique was to work, it could be implanted in humans. So that would be the main advantage as opposed to the genetically encoded sensors. Yeah. Yeah, I don't know. Who wants?
Which seems to be an animal, which is great. I'm curious how, if you could reflect on what is the range you need for a biological significance? Is it in that range already, or is it?
Yeah, that's a good question. So what you were asking about the sensitivity, if the detection limit is enough, like the sensitivity is enough basically to detect things in vivo or ex vivo. For serotonin, actually, we have the good range, and this is basically what I put here are the ranges that they have detected with the genetically encoded sensors. But the issue, I mean, for dopamine, if you look at the traces that I showed, the 500 nanomolar to 1 micromolar, and this is usually, I mean, up to probably a maximum of 8 micromolar is the range of that they have detected for dopamine. So we need to improve that signal. It is, I mean, arguably a good signal, but what I need to do to improve it is to increase the laser power. And if you increase the laser power, you induce photobleaching of the dyes. So there is a balance to achieve. So eventually what we want is really to get the better aptamer. Yeah. To like more get this signal, but for 500 nanomolar. We are a little off by like an order of magnitude, let's say. Yeah. For serotonin, we're good, but the issue with serotonin is that we don't have a reference technique to benchmark ourselves with, like a well-established technique. There are techniques that are like controversial or arguable. I don't know, like neuroscience is like a bit of a controversial like field, but yeah, we needed a benchmark, and that's why we picked dopamine first, because FSCV is roughly well established for that.
Are there limitations in the types of molecules that can be detected here? Like I'm thinking, talked a lot about some of the small molecule neurotransmitters, but what about some of the lipophilic types? Will it work similarly?
Yeah, I have not. Yeah, this is what we could do is actually modify what people in the lab. So yeah, what he asked was, what about the lipophilic type of molecules? Like we talked about these small molecules, but could we generalize it to lipophilic type molecules? And it's a bit hard because of the structure of the DNA and the limitation with it. But what people have been doing, and Larry Gold, I think, is like a big like pioneer in modified aptamers with like other moieties, like non-natural aptamer. And that's like if you look up Somalogic is like a company that they pioneered to actually make modified aptamers. So instead of the DNA backbone, they would actually modify it with different molecules, like other molecules and other moieties, to make it a bit like maybe more hydrophilic or hydrophobic and act like a protein. So mimic the binding sites of like antibodies or other like peptides. They're trying to modify the DNA, but it's challenging. We've tried it in the lab and it hasn't worked out well. But that's the only thing I know about it. But it is challenging, yeah. So you're asking whether we know how basically the aptamer is binding to the target, like the structural features of the binding site. Is that it? Unfortunately, the techniques to crystallography have not been successful for many aptamers. They've been successful for one, I think, or two, and these are RNA aptamers, not DNA-based ones. So it's been super challenging to do that for aptamers. So what people are trying now is to use machine learning and AI. I guess like everybody is trying it, but this has been actually helpful for aptamers. So what they can do is get all the data from the selection process and map what are the good binders and map their sequences and see energy landscape and try to reverse like engineer and try to understand what could be the binding site. But it's all indirect so far. There hasn't been a method that could help us visualize how the aptamer is binding to the target. Yeah. There is like Oxford Dynamics, something like that. I forgot the name. There are many tools, but just for DNA, for the DNA structure, like they have used them to build like these higher order structures with DNA, like a smiley face or like a robot, but they haven't tested it in interaction with a target. Like how they're trying to understand basically, is the binding site formed before the target binds? So the target is sensitizing the formation of the binding site, or when the target binds, it actually folds around it. Like we don't understand anything about how the aptamer binds, and that's difficult to simulate in things like Oxford dynamic. I'm inventing a name. It's Oxford something. I don't know. Don't take my word on it. Yeah.
Curious, I'm not like the neuromodulators, so they're like other assumption is the problem is somewhere similar. So when you do your selection, if you're looking for something for a particular neuromodulator, do you perform counter selection?
Exactly. Yes. Yeah. So we do actually, is asking whether we perform like counter selection to be able to distinguish neuromodulators that have very similar structures, and that's dopamine and norepinephrine. So yeah, you do like a negative selection with norepinephrine. So you don't select the ones that are binding to norepinephrine. This is very challenging, but what people are trying to do is like now just measure in regions where they know it's mostly dopamine, like the concentration of norepinephrine is much lower. So they get around that problem, but it is challenging to do that. And it does require lots of negative selections. Yeah.
I have another question from a Zoom.
Yeah.
They said, it sounds like this could be modified to detect any type of organic molecule. Do you think these could be used to monitor the chemical environment of brain tumors?
Definitely. I mean, that's the goal, is to really expand it to other applications like cancer. Yeah, the only problem for now, and I think I showed at the end, like the cortisol data, for now we're able to measure in whole human plasma, but the blood is a bit more challenging to get things to be specific, to get fluorescence to work in like the blood environment or in a tissue environment. This is what we're actually trying to optimize, to protect these aptamers and the fluorophores from the tissue environment, the moieties that could quench the fluorescent signal. So that's what we're at, but it definitely could be expanded to other applications. Yeah.
I had a quick question on the temporal, on the x-axis. So you mentioned a lot about that and cyclic photometry, and just kind of compare. I mean, they're different, but the applications would be similar in some areas of behavioral neuroscience, like the mouse, and you're trying to track behavior. Like, what are those individual data points? Like, what is the resolution temporally?
Oh,
Like fast. That's like fast scans kind of continue.
yeah.
But you're saying there's a limit.
So here, I think we can go down to 50 milliseconds.
Oh, okay.
Yeah, so it's pretty fast actually, and we can actually bin it and get it to be faster. So the single photon counting module are pretty fast. It's just like for us, we want to limit the amount of data. So we want the computer not to crash for long-term measurements. So we try to bin it so that we collect every 50 milliseconds a data point, but usually it could be faster than that too. Yeah. But you're limited by the aptamer kinetics too. So if you see cortisol is like instantaneous, you cannot see the in-between kinetics. The moment we add it, we see the maximum signal. So it's a very fast aptamer, but dopamine has given us like a trouble, because you see that you don't reach equilibrium within like the milliseconds that we are measuring. It's a bit slower as an aptamer. the K on is slower. So you could be limited by the aptamer's kinetics. Yeah, in this case.
Could you track, like, I know this is still the beginnings of it, like, again, comparing like with fast scan, like those moment-to-moment things, and things of interest to like with, addiction or something, like here's the mouse and it's approaching the lever, and it's a lever that they've been responding on previously to get cocaine, but as they even approach it, like Paul Phillips' lab in Seattle, you see a surge in dopamine, like, would it then be stuck there too long to then see what happens next, like the off?
It could be in this case because we do have like with the dopamine, the serotonin, if you look at it, it's super fast. Like we're here, it's just like a manual thing to wash the chamber. Like the serotonin is stuck in the chamber and we're not able to wash it completely, but the switch is super fast. But the dopamine is a little slower. So you see here, like on the time axis, you're not able to see it, but if I actually zoom in, it's actually in the seconds, and you want it to be sub-seconds, yeah, to capture all the dynamics. Oh, it's not perfect. Yeah.
I'm wondering if there's been any consideration or work towards creating some sort of a feedback loop or whatever, where there's something detected here that triggers the release of more drug or whatever it is that you're investigating or you want to deliver. That is just kind of this automatic kind of response to a change in this, physiology that you're detecting here. Is that something that you are interested in, or do you see this going? I can almost see it like a drug delivery device where, it's circulating throughout the body. There's some sort of signal that maybe like tumor gives off or whatever that triggers the release of the drug to that specific location. Is that possible with this system? Is the technology there to do something like that?
I'm hoping, because my lab will be focusing on that. So hopefully it would. So that's what I mean, basically, this is more like science fiction like image, but eventually what we want to do is build like these structures and functionalize them with the aptamers, so that when they sense something, they open up and deliver stuff. So this is the goal, to build like these multifunctional nano machines or nano robots. It's going to be very challenging. Like you're dealing with a very complex environment, and putting multiple aptamers into one entity, we need to understand how their cooperativity works in a like nano robot, like made of DNA or made of polymers. We need to understand how they interact with the system. So that's why like it's going to take a bit of studies, but that's the goal of my vision now, is to take it to the nano. Exactly. Yeah. Yeah.
One more question. Is there at UBC, is there something analogous, or I guess just similar to Stanford, the precision medicine kind of a emphasis or group?
There is the brain center, where many people are working on, like I think they're from the Department of Medicine, Engineering, from different departments, some from the psychiatry, some from pharmacology and anesthesiology, I guess, and they want to work on like the brain in general, and some more specifically on biomedical devices for neurodegenerative diseases, like, and trying to detect like molecules like that, either in the blood, actually dopamine, or other neuromodulators in the blood, not only in the brain. They do want to understand the effect of drugs, antipsychotic.
Psychotic.
Sorry, yeah.
I want the psychedelic.
Yeah, yeah, exactly.
So knowing what the appropriate levels are for treatment, so therapeutic.
Yeah, because they do have side effects, like for the cardiac diseases and type 2 diabetes. I guess they do cause some unwanted effects, and they want to study like the doses for precision medicine, basically. Yeah. So there is a department that I started talking to actually start this also at UBC. Yeah.
Any other questions? Good. Well, we'll be talking more later today, but thank you so much for.
No, thank
coming.
you.
For anyone, especially students, but anyone, please hang out afterwards. There's cookies and coffee. Ask questions. It could be very detailed about the chemistry and all that, but also, why did you get into this field? I mean, who knows? You might learn something about what you might want to do someday. And thanks to, thanks to the alumni. Sorry, I'm speaking into my microphone. The people out in the
Oh, yeah.
groups here. Thanks for streaming this on Zoom. Very cool for our people in the Zoomiverse. And I'll,
Sorry about the next slide thing.
oh,
I ruined the dopamine circuitry for them.
oh. They'll have to read up on it. Okay, well, one more hand for Dr. Hurts.
Yeah, thank you. So should I stop?